Okay, in this session, we will be understanding the cell-mediated cytolysis. So, first, what is a cytotoxic T cell? Right? So, cytotoxic T cells, also known as the killer T cell, is a type of white blood cell that plays a central role in immune system. Okay, these cells are part of the adaptive immune response, which is the body-specific defense mechanism against infection and abnormal cells. For example, cancerous cells. So, in this effector CTL generation from the CTL precursor. The CTL, that means the cytotoxic T lymphocytes. So, in simpler term, naive T cells, okay, also called as a CTL precursor because it can't kill target cell because they have functionally immature, okay, and they become effector killers, effective killer cells known as the CTLs after activation. So the cell differentiate into the functional CTL with the cytotoxic activity. So in this picture, first, this is the immature CD4 plus T cell. Okay, and here it is the immature CD8 plus T cells. You can also say the naive T cells. Okay, TC cells. Now this is the antigen presenting cell, and this is the T cell receptor. Here it is also T cell receptor. MSC2 and MSC1. Okay, MSC2 with the antigen, MSC1 with the antigen. Once they binds and then activated and they differentiate into the mature helper T cell. The types of TH1, subtypes of T helper cell, TH1 and TH2. Here it is the once they binds with this receptor, T cell receptor, MSC class 1 molecules, and they activated into the CD8 plus, okay, mature cytotoxic T cell. So this further this activates CTLs from CTL PS. Okay. So antigen presenting cells. Okay, refers to antigen presenting cell that is undergoing process of activation of lysosome, which enables it to effectively stimulate and interact with the T cells, either cytotoxic T cell and helper T cells. Okay. So CTLPS, that means now CTL, okay, mature into cytotoxic cell, they need to interact with the lyson APS. Okay, so lysoning of APS, that is the antigen presenting cell, they may be macrophages, then B cells or dendritic cells. Okay, so here we take the example, for example, dendritic cells. Okay? So the dendritic cells, okay, first they must be licensed, or you can see the activated in several ways. For example, first CD4 plus helper T cells, TH of the TH1 or TH17 subtype. Okay, this one binds to this MSC class 2. Second, engagement with the microbial product. Here is a bacterium, okay, and they having the toll-like receptor, which activate toll-like receptor. Now this is the how APC, okay, activated. We take the one of the example. For example, this is the macrophages, okay, and bacteria engulfed by macrophages encased in the vacuoles, okay, vacuoles. And second step, it is the lysosomes fuse with the vacuole and digest the entire bacterium content. And then the antigen, okay, antigen MSC class 2 molecules binds with the antigen, okay, third step. Antigen from digestive material, bacterium are presented with the MSC2 on the cell surface. So the how MSC2 molecules are expressed on the macrophage surface, cell surface. Now there is the important generation of effector CTLs. So the how effector CTLs are generated from the CTL precursor. So for differentiation of naive CD8T, okay, that means precursor CTL into functional CTL that requires multiple of events. So first precursor CTL, must engage with the class 1 molecules. Okay, second. License, once the APC licensed, okay, then they express the class MSC1, okay, class 1 MSC molecules. So the must CTL binds to the MSC peptide, class 1 peptides. Okay, and co-stimulatory ligands on licensed antigen presenting cell, CD80, 86 and CD28, which is also binds with them. Now, the licensing dendritic cell occur either through the engagement with the activated CD40L helper T cell, TH1, TH17, or through the engagement with the pathogen. Here is also pathogen toll-like receptor. Okay, so for CD4, it's required for the proliferation and memory generation. Okay, and the CD40 T cell help can given prior to CTL engagement of the dendritic cells. Okay, that is also helpful to the how CD4 plus help to binds to with the CTL to the MSC class 1 molecules. They also secrete the cytokinin, okay, interleukin 2, itself null cells. They also, once they activated, they binds to the MSC class 1 and they secrete the interleukin, which is the interleukin required for the proliferation and differentiation of the CTL. So this is simultaneously, okay, one APC parencytic cells, they express the both MSC class 2 here because CD4 plus, okay, CD4 plus T cell receptor always binds to the MSC class 2 molecules peptide, and second one is it is the presenting another class 1 MSC peptide which is also binds with the null CD8 plus T cells, okay, cytotoxic T cell with their T cell receptor. So this is a simultaneously event. Okay, first the MSC class 1, MSC class 2 peptides interact with the T cell receptor and they produce the interleukin which is helpful for the CTL generation, proliferation or differentiation. Okay, this is also cytokine interleukin 12 also produced by the APC molecules and itself, okay, CD8 plus T cell cytotoxic T lymphocyte also produces interleukin itself. So this is the how CTL, okay, effector CTL generation from the CTL periods. Okay, so here it is the sum of the multiple choice question. Okay, question 1, question 2. Carefully read and solve these questions. Kindly solve each questions and share your answer in the comment section below. Thank you for watching.